Okay, last but not least, Dr. Christine Georgakakos from SUNY ESF. Okay, Christine.
Thanks for hanging on, everybody. I know we've been sitting down for a little while and are trying to go to lunch, so thanks for continuing to engage with us. First, I want to say this is a big project. Bahram and Greg and everyone are all involved, so hopefully we can eventually tie all the data that we'll get into those ones. The folks that are most acutely involved in this one are Rakesh, Dominique, and Steve, so if you're having lunch with any of them and have questions, feel free to ask them as well. I think everybody is equally informed about what I'm going to talk about. So when we think about Skaneateles, I think we think a lot about our recreation users. We think a lot about inputs in terms of surface water. We've heard a lot about atmospheric deposition today. But I want to thank the person who asked the question about what are those other inputs? What are the septic users doing here? And that's sort of what we're trying to get at with this one. They're sort of an interesting anomaly. You could consider them a point source. You could consider them a non-point source. A lot of these systems have been there for a long time. Some of them are newer. There's a lot of diversity in how they were installed and where they are in the landscape. And so our question here is really, what are those have really high septic density? Are they sort of homogeneous around the lake? Is it controlled by other landscape scale factors, that kind of thing? And I will go ahead and say that I'm not going to share any data today. We're still in the process of analyzing and collecting this stuff. So again, during lunch, if you have ideas for us, happy to engage in those types of things as well. I want to thank Dr. Lewis McCaffrey, who created this map a little while ago. Thanks for sharing it online so we can use it here. This is a summary of where we are in the watershed. So the colors represent Skaneateles watershed. The reds are those areas of higher septic density. The greens would be those areas of lower septic density. You'll notice that things are not necessarily homogeneous. So if we sort of draw a line in the middle of the lake, we have those more areas of higher septic density on the northern end of the lake, with some areas, this is the town of Scott down here in the bottom, those more rural spaces that don't have that consolidated wastewater treatment, but do have a lot of residences. You'll see high septic density there as well. But I also want to draw your attention to the diversity as you move in towards the lake and away from the lake. We have really high septic density right on the lake shore, and that density decreases the farther away we go. That also is controlled a little bit by the geology of the system. Down on the southern end of the lake, we have really steep walls of our lake, so you can have one house wide away from the lake, whereas on the northern shore, you can have sort of more homes closer together, closer to the lake, and so that's part of why you see this really high septic density up in the northern end, and that lower density, though you'll still see some of those sort of a solid black line all the way around the lake, right? We enjoy that lake shore living, and so that's where this stuff is coming from. I'll also note that these dots are at the center of the tax parcel. So this is a publicly available data set. So anybody who noted that they have on-site wastewater treatment is noted here at the center of the tax parcel. These dots do not represent exactly where those septics are located. Those are a little more nuanced. I'll talk a little bit about that in a second. So if we take a step back to just sort of understand what is happening in on-site wastewater treatment, whether your home is located right on the lake shore like this one or farther away, most of those are pumping their wastewater to a septic tank that is a little bit farther away from the lake, allowing any of the contaminants that are in that wastewater to be microbial degraded within the tank, within the leach field, in the soils around the leach field before it hits any sort of surface water. So in an ideal functionality, you have very limited nutrient or contaminant transport because the soil is acting as your filter. Similar in reverse order as you would think of it as a groundwater well. The soil is filtering out any contaminants that we might not want to go anywhere. If you think a little bit about different types of contaminants, that soil is going to act as a more effective filter for different types of contaminants. So sort of like Ryan was mentioning earlier, phosphorus tends to bind with sediment particles. So hopefully those soils are acting to filter out a lot of the phosphorus before it reaches our lake from these septic systems. Whereas if you're thinking a little bit more about contaminants like nitrates, nitrates tend to be more dissolved in solution. Those ones, the soils are going to less effectively filter them out, but could be microbial degraded instead. So depending on the contaminant that you're thinking about, it really matters. Is the soil going to effectively filter those things out of the system? Phosphorus and nitrogen are one that we talk a lot about with algal blooms, with Skaneateles Lake, but I'm going to go ahead and say that there's a lot of other contaminants going in here. Frank mentioned this at the beginning. My group thinks a lot about pharmaceuticals and personal care products and other cleaning detergents and things like that. And each of those also functions in different ways. So some of those act more like phosphorus, and they're going to absorb really strongly to your soil particles. Some of those act more like nitrogen, and so they're going to be passed a little bit more easily through these systems. Our goal here is to try to understand, are these septics, are our nearshore inputs higher in these areas that have a lot of septics, or do we see sort of similar patterns around the lake? So to try to answer this question, we've input a couple of different monitoring stations. One is a shallow surface water well right at the edge of the lake. So before any water that's coming off of your landscape in the subsurface hits the lake, we'll take a sample and see what's in that sample. We're going to look for a whole bunch of different things. I'll show you what those things are in a little bit. And then we'll also take a near shore lake sample. So if we see really elevated stuff in the groundwater samples, do we also see that right near the lake shore? Or is it diluted enough so that we don't sort of see those in the detection methods that we're using? The other question, though, is about the filtration of the soil, the filtration capacity of the soil in these systems. So what we really need to know is how effective are they, how many of these contaminants are being held in those soils right before it hits the lake as well. So we also have, sorry, I'm holding too many things. We also have two other sampling sites here. One is a subsurface sample where the groundwater table will be, and the other is a surface soil sample. And we'll extract a bunch of different contaminants from each of those things as well, try to see if we see at the groundwater table, do we see really elevated levels of phosphorus? Do we see that you can absorb more phosphorus onto those soil particles? And then same thing for some of those other pharmaceuticals and personal care products. Do we see that those soils are already sort of saturated? Are they at their extent of absorption potential, or can they continue to take on these things? For those of you who engage with septic tanks around Skaneateles Lake, a lot of these septics are very old, right? They've been in place for a long time. They've been fixed and added and adjusted to suit the needs of our current homes. But the systems and the places where they are the same. And so the question sort of becomes, have we saturated those soils? And is that level of saturation enough before it hits the lake? Or do we still have that sort of buffer zone or far enough away before any of these things get down there? So we're trying to take this sort of dual approach, water samples, soil samples, to try to address this question. What does it look like? What if you were to input one of these wells, and thanks to those of you who are online or in person that have one of these wells in your residences, we dig a nearshore well down until we hit the groundwater table. So the idea is that we want to make sure that that well is wet when we want to go sample. In general, that's something like 2 to 4 feet if it's right near the lake shore. I will say that the Skaneateles Lake water level is a little bit complicating of our analysis here. Sometimes we dug a well and then the lake level drops and our well no longer has any water in it. So we're adjusting some of those techniques to try to make sure that we can get samples all year round. We drop a PVC perforated pipe down in there with a water level sensor so we can figure out what is the depth of the water table throughout the year. It'll be continuously monitored. And then we install this protective casing around it so that, number one, our well doesn't move around too much, and then also so that people don't step in it and hurt themselves as they're engaging with the lake in the ways that they're recreationally or otherwise. I will put a plug that we're looking for other folks to install some of these things. So if you're jazzed by this and want to engage, please chat with any of us at the beginning who are involved in this project. And I will say that we're looking at a whole slew of different contaminants here. So those primary ones, nitrogen and phosphorus, are the ones that we usually think about with algal blooms and stuff like that. But I will say that these pharmaceuticals and other products they have many functions. So antibiotics, though they are intended to interact primarily with bacteria, they also have interesting effects on other organisms like aquatic macroinvertebrates. They increase stresses on larger fauna such as fish and things like that as well. So those untargeted byproducts are interesting in terms of what's happening to the ecosystems of the lakes that we're in. But then also other things like artificial sweeteners are also antibiotics. And so we use these things for one purpose, and then they enter our ecosystems and do something else and do those changes and engage with our algal communities, like you've heard everybody sort of talking about today. Are those some of the triggers that turn on or turn off which populations are more able to adjust and modify their life stages based on the stressors that they're experiencing? Paired with this also, we'll do some fecal indicator bacteria analysis and some antibiotic resistance gene stuff. So trying to pull in some of those more biotic factors as well as the abiotic stuff. And then we have a whole slew of water quality metadata that we're taking alongside. So with that, I'm happy to take, hopefully this was pretty short. People can ask as many questions as you want. Again, if you know folks who are excited to have a small well installed on their residence or a place close to, and it can be have a septic, it can be on a sewer shed, it can be any of those diversity of wastewater treatment that we have. And then lastly, I'll just give a shout out to all the undergrads, both at ESF and at SU, who've been helping collect all this stuff in addition to the folks on that front page. Yeah, so hopefully I have time for some questions.
Thank you for your presentation, Christine. I'm curious, are you considering looking at any dependencies between your observations and the condition of the upstream septic system, maybe the age of the septic system or the style of septic system?
Yeah, absolutely. Great question. Yes, we're trying to get all of that data. So if I will say that the diversity of that data that the residences have is not consistent. So any of that stuff that is available, we are open to. So we're trying to get a whole range of septic systems across ages. So think about a very new septic system probably works very differently than a septic system that's been in place for 50 to 100 years. We have 7 sites so far that span anywhere from, I think 10 years is our youngest, to over 50 years. So anywhere in there is what we're looking for. We also have a pretty diverse range of the types of systems. So a bunch of these are pretty old systems that are designed to be leaky. So they're not necessarily sealed tanks, but they're supposed to sort of like emit into the landscape a little bit. Whereas some of the other ones are more like PVC piped style perforated pipes that go into a built up leach field that are then going to engage in the soil microbiology in a pretty different way. We're looking for all of those things. So trying to get sort of the whole spectrum of different types of wastewater treatment that happens on site. I will say that Skaneateles Lake has a pretty diverse range there. There's a lot of folks that do more cesspools, so collect wastewater but do not treat on site. It's pumped out and treated elsewhere. There's also things like composting toilets and stuff like that, as well as these a couple of homes contributing to a single septic tank and then distributing to in a larger system like that, depending on what's available in terms of landscape. So yeah, there's a huge diversity of wastewater treatment stuff that happens, and I'll say that we're excited about all of them, so don't hold back if you know folks that are willing to engage. Yeah, Liam.
Hi, Christine. I was wondering if you have done any sort of kind of back of the paper napkin math on if it is reasonable to assume that the soils are fully saturated with phosphorus. I'm kind of imagining the soils as sort of like a sponge, and you're having your leachate field slowly saturate that sponge. Given, we're looking at probably 100 years or so of length for some of these sites, is it reasonable to even assume over a certain volume of soil that they are fully saturated or not?
Great question. I don't know the answer to that. What I would propose is that I think we should do like a phosphorus addition experiment to see how much more can the soils hold. I will say that a lot of our lakeshore sediments are very gravelly. So think about like large particle style soils that are often washed down off the landscape. Those types of soils don't have very many adsorption sites. So something like smaller sediment particles should have more adsorption sites. So it's going to be really soil specific. And we definitely need to do that calculation in the form of a lab experiment to figure out what's going on. Unless somebody else here has done that and would like to share. Cool, thanks.
Just a few announcements for our folks online. That does conclude our online session for the morning. We will be moving into lunch and then breakout sessions with folks here at the conference. But wanted to thank Christopher Bakura and Molly Schultz, which have been part of the technical team, making sure that the online experience has been as great as it is. So thanks a lot to ESF and all of that, and Steve again, and Charlie. I did put into the chat a few upcoming events. We're loving this science. It's been jam-packed today. There are some events coming up that I will make sure that there'll be follow-up on this conference, this summit, with info about the recordings. We'll be connecting with the presenters on what they'd like to share with their slide decks. but get more information out to all the registrants that have been part of this. Wanted to say thank you to all of our presenters and everybody that's participated with the wonderful questions today too. But the events coming up, the first one is the Bob Brower Symposium that the Owasco Watershed Lake Association hosts. It's Saturday, March 14th from 9 to noon. Great event. Highly recommend that. And then there is the New York State Federation of Lake Associations annual conference that is on May 8th and 9th up in Lake George. Actually the first one coming up, I'll back that up, January 29th at the Finger Lakes Institute at Hobart William Smith is the Finger Lakes Research Conference that's coming up very soon. Early bird deadline's January 15th. And then the other one that Steve's been a great part of is the fourth annual meeting for the Healthy Waters Solutions. And that will be May 19th through the 21st. Registration's not open for that, but if there are folks that are interested in presenting, the deadline is tomorrow for proposals. So that's all I have for those announcements. Thanks again. Enjoy lunch. Looking forward to more discussions. And thanks for everyone who participated online.
Thanks again to everyone presenting. Thanks for the participation. We'll meet back here at, we'll say 1:05, 40 minutes, to do breakout sessions this afternoon.
Just a note on all of the eating ware that you're using. Everything should be compostable if you got it here, except for the plastic cups, which are recyclable. If you got your stuff somewhere else, then look close and see if it's compostable or not.